Okay, so I started recording the meeting. So welcome everyone to this Friday Population Dynamics meeting. So today we have Nicolas Desprat, and I will share my screen to introduce the speaker, if I actually can start from the first slide. So Nicolas Desprat from the École Normale Supérieure in France, in Paris, and the title of this talk will be Multiscale Organization of Rod-Shaped Bacterial Microcolonies. Some word regarding the past experience and the research of Nicolas. He got a master's degree in solid state physics and condensed matter at the University Sorbonne. Then I got a PhD at the University of Paris in single cell rheology, and I did a postdoc at the Curie Institute. To become after associate professor at the University of Paris regarding physics of microbes. And the goal that usually he has is to understand how physical constraints shape microbial communities. So large part of his research is on that. And also dedicated to bacterial communities and host pathogen interaction. Specifically, he does studies on collective behaviors in Chlamydomonas renarti, and then on Saccharomyces cerevisiae in fluctuating environments, and also something regarding evolution in a spatially structured environment. So today, as I told you, we will chat about the multi-scale organization of rod-shaped bacterial microcolonies. And so with this one, I would like to thanks Nicola to being here. I will stop sharing my screen, and people can actually interact with the speaker today by you can raise your hand. There are reaction here on the bottom, or you can write on the chat. But the point is that you can during the talk interrupt and create a discussion. The talk will last maximum 1 hour of the meeting. And again, so thank Nicola to be here, and the screen is yours. So you can share your part. Thank you.
Thank you, Marco, for the introduction. Hello, everyone. So thanks also for the invitation. So I'm going to share my screen. So do you see it?
We see the full screen, yes.
Okay, perfect. So I didn't hear you very well, Marco. So if you don't hear me, tell it to me.
Yeah, actually, we can hear you perfectly. So maybe my microphone is not good.
Okay. Okay, so I won't talk today about algae. Neither I'm going to talk about yeast. And so today I'm going to talk about bacteria and how the different scales of the organization, meaning from a molecular scale to the population scale, are coupled together. Okay. And so this is a work that has been done mainly by Dario Dell'Arciprete, Simone Guette-Marquet, and also Marie-Cécilia Duvernois for the mechanics part. So, so first of all, as you may know, the bacteria they are not freely floating in space. Okay, so they interact with a physical and chemical environment, and they also interact with other species. So how do they achieve that? They achieve that by the means of outer membrane proteins. Okay, and so there are mainly two types of those proteins. So either we have from one side here flagella, pili, adhesins. So these these surface appendages they mediate mechanical interactions, and so basically they are responsible for motility, swimming, swarming, or twitching or they are also responsible for host pathogen interactions for they trigger the adhesion to the host cells, or more generally, they can serve to form biofilms. And so on the other side, we have the chemical interactions. And those chemical interactions, they are mediated by efflux and import pumps. And so, first of all, they are used to uptake nutrients. But not only, they can be used for quorum sensing signal to secrete them and uptake them, and also for toxin secretion in bacterial competition and many other aspects. And and so and in dense phase, what we observe for bacterial communities is we can observe collective behaviors. So here I picked up two examples from the literature, one from our group, which is the example of pyoverdine that is secreted by Pseudomonas aeruginosa, and the other is the chemoelectrical signaling in Bacillus subtilis biofilms. Okay. So on the left, what you see is what you see in cyan is the pyoverdine. So pyoverdine is an iron chelator, and it is secreted in the environment in order for cells to uptake iron from the surrounding environment, because iron is essential. Okay, and as you can see, there is a non-trivial dynamics inside the colony. On, on the right, do you see a macroscopic biofilm of Bacillus subtilis? What is stained here in cyan is the thioflavine, and so the thioflavine reports for the, the membrane potential of the cell. So this is an Nernstian dye. And so what you see here is that there are oscillations, and those oscillations are made to couple the exterior and the interior of the biofilm in order for the interior cells to be able to grow. And so in those two systems, what is remarkable is that cell-cell contacts are essential. So in our system, what we measured is, so here you have on this axis, you have here the concentration of siderophore within a cell, C, and the concentration of siderophores in neighboring cells. And you see that if a cell is more concentrated than neighboring cells, basically the cells will likely to release siderophores toward the neighbors. And on the contrary, if the cells is less concentrated than its neighbors, it will uptake siderophore from its neighbors. And we showed in the system that if a cell is importing from its neighbors, okay, so meaning that the neighbors here are more fluorescent than the cell itself, there is a higher growth rate than than if cells are less concentrated than their neighbors. So clearly here, the local exchange between bacteria, they directly impact the fitness. And so, shit. Are you still online?
Yes, we are, but probably we lost
Not probably.
the full screen.
We lost it. Shit. You get it?
Yep.
Yeah, it's back. Thanks.
Okay. And so, so in this system, the the electric signal is conveyed from cell to cells also by means of direct cell-cell contact interactions. So that's why we came to this question. So why, starting uh, from bacteria that are rod-shaped, so basically they are spherocylinders, why the microcolony does not reflect the the geometry of its constituents, and why the geometry of the microcolony is more or less round. Okay, and so this could seem to be a naive question, but in this case, you privilege the interaction with the environment. So this is a scenario that will privilege individual selection, and on the contrary, here the interaction with neighboring cells and siblings are maximized. So it will privilege the scenario of group selection. So in order to investigate this question, what we did is we grow bacteria between a cover slip and an agar pad. And so that's what you see here. The arrows are just depicting the bacterial orientation from the old pole to the new pole. And you see that at some point, there are no more arrows in the center. This is because there is a double layer formation. So, first, uh, we investigated the role of adhesion in these dynamics, and we saw that if we knock down proteins that are used to either synthesize polysaccharide, or if we knock down proteins that mediate surface bacteria adhesion, what we see is that microcolony gets more elongated. And regarding the formation of the double layer, we can question at which size of the microcolony these double layers arises. And what we saw is that, again, if adhesion gets lower, then microcolony can expand more on 2D before forming this double layer. Okay. So then after that, we measured the forces thanks to deformable substrate. Okay. And and we and what we observe is that we observe adhesion foci that are transient, meaning that cells are they form adhesion and they also grow, and so meaning that when they grow in the center, they move all the cells away, and so they can break adhesion of neighboring cells. And so thanks to that, what we can relate is the force that we measure at the adhesion foci and the size at which the transition from a monolayer to a double layer microcolony occurs. And we observe that there is a negative correlation. And indeed, there is a negative correlation because the nice picture is that the more neighbors you have, the more difficult it is to push all the neighbors. And at some point, it becomes favorable to deform the gel which is above. Okay. And so at the end, I won't present that, but we can have an analytical expression for this curve, which is not exactly a straight line, by just comparing the adhesion which is necessary to increase the elastic energy and the repulsive energy on the plane compared to the energy that is required to deform the gel. So, so after that, we wanted to, so we wanted to know whether the environment could modulate the strength of the adhesion. And so we came back with Pseudomonas aeruginosa and siderophore production, and And so in this system, we can either use a control medium or we can use a medium for which we add a human iron chelator, the transferrin, uh, which is a very strong chelator that will compete with pyoverdine. And so in this condition, pyoverdine secretion is essential for growth. And cells are also a bit stressed. And what we see is that when we reduce the level of iron, the saturation forces gets stronger. So clearly, it indicates that adhesion responds to the environment. So now to be able to really design a mechanical model of microcolony growth, we have to know what are how the adhesins are spatially distributed in single cells. So therefore, we devised a very simple assay in which we measure two things. The first thing that we measure is the displacement of the center of mass of the cell. And the second thing is the elongation of the cell. And so we have this delta X and this delta L. And, and if, and we know from the literature that the synthesis of the cell wall is homogeneous along the cell. And, and, um, and so in this scenario, if adhesions are also uniformly distributed, what we expect is that cells will elongate around its center of mass, and the center of mass will remain at a fixed position. On the contrary, if adhesions are located at one of or the other pole, the old or the new pole, the insertion of new cell wall material will translate into a displacement of the center of mass. Okay, and so the relation should yield a linear relation, and so the slope should be positive if adhesion is at the old pole, and that's the convention we took, and negative if it is at the new pole. So we perform laser ablation in order to identify the old pole and the new pole, and we found that indeed cells, they do not homogeneously adhere to their substrate, and they are more strongly attached to one pole, and they are more strongly attached to the old pole. Okay. So, and here the two curves, in order to rule out artifacts from the ablation, we performed either ablation systematically on one of the daughter cells, or we alternated between the two daughter cells. And you see that the results are the same regardless of the way we did the ablation. So,
Yes, yes.
okay, sorry.
Perfectly.
Okay, so so now that we know the macroscopic dynamics of adhesive forces, and we know how they are microscopically distributed in single cells, we we can design a very simple mechanical model for the morphogenesis, the microcolony growth. And we can measure some parameters. And one of the key parameters is going to be basically the nematic order parameter. And so in red, here you have the experiment. In green, you have the simulation. So, and those simulation, they correspond to a case in which we put adhesins only at the poles. And we compared this green case with the gray case. And in this case, adhesins are distributed homogeneously on the surface of the bacteria. And what we see here is that the the bacteria, they remain more aligned, and which results in a more elongated microcolony. So clearly, the fact that microcolony gets round comes from the fact that adhesion is polar, meaning that adhesion is located at is located at cell poles. And how do we understand that? We understand that simply is that the fact that adhesion are tethered at the pole, it will create pivots with the substrate. And actually, when bacteria, when they grow, there will be there will be a tension in the system, and the tension will be released by rotation. Okay, and so these accounts for the first reorganization, going from two daughter cells to two daughter cells that are close together, and so on. So,
Question? Sorry,
ouais,
I have a question.
yeah.
What are these adhesion foci actually, for example, in E. coli? Are they particular proteins which bind to the substrate or protein complexes or something?
There are there are plenty of proteins. So so here, for instance,
You said, yeah, okay.
so there is a delta 4 adenosine. So sorry, I made this talk more
No, it's good.
physics talks than biology talks.
I didn't never heard about this in E. coli, so I'm interested to know that they have.
So first of all, the flagella, it is mainly used to swim, but it can also stick to the surface. I mean, all the experiment of all the Berg's experiment are using the fact that the flagellin, it binds to the to the surface. So then there are the type 1 the type 1 fimbriae, which looks like a bit like pili, okay, but which are not pili. And those fimbriae, they have a lectin at the very end. So, and the lectin, this is this is sugar. It has an affinity with it is like a sugar, and it has affinity with no, it has an affinity with sugar. For instance, mannose or something like this.
Okay.
Okay, and there are many, many other systems. I'm going to talk specifically of one of them, which is the AG43, which is responsible for the cell-cell adhesion. Okay, but you have also curly, and you have a lot, and this delta 4 pole is for polysaccharides. So here, polysaccharides, these are not strictly speaking proteins. This is these are extra polymers. These are extracellular polymers that are secreted. And basically, these are when you have polysaccharides, this is sugar. And sugar, do you know that if you have a syrup, it will stick. And it's the same for bacteria.
Thanks, that's helpful.
Okay. And they have, but they have many, many, many adhesion systems. And so far, no one succeeded in designing a bacteria or a surface that, that on which bacteria cannot adhere. Okay, so so what we saw is that the level of cooperation between cells is actually organized by the fact that adhesion are polar. So now what we want to understand is what are the determinants for the cell polarity. And so so when an outer membrane proteins, before an outer membrane proteins gets exposed on the cell surface, it should be first synthesized or expressed in the cytoplasm. Okay, So then once it has been expressed, so basically expression, transcription, translation, then it should be secreted into this space, okay, which is the periplasmic space. And then in this periplasm, it diffuses, okay, before it is inserted on the outer membrane, okay? And so, so here I'm gonna focus on only on gram-negative bacteria. So gram-negative bacteria. So sorry, I forgot to tell you, they have two membranes. So an inner membrane and an outer membrane. Okay. And so what is not depicted here is the cell wall. Okay. So here you should think this space as a very crowded environment with with a reticulated. So, so what do we know from the literature of the three main steps? Okay, so first the translocation, diffusion, and exposition and insertion. So, do we know that one of them has displays a bias or not? And the answer is no. Okay, so if we took images from the literature, what we see is that the here, so sec is a protein complex that that is this is not the only one, but for the protein we are looking at, this is the main one. So and sec is basically a channel that takes protein with a certain peptide signal. Okay, so it means that the protein at its N-terminal region should have a specific sequence that is recognized by this transporter, and then this this channel allows proteins to be shuttled from the cytoplasm to the periplasm. Okay, and this is unidirectional. It cannot go the other way around. Okay. Also something which is important here to mention, is that the secretion of proteins are co-traductional. Okay, so meaning that there is no accumulation of proteins in the cytoplasm. Okay, so sec is distributed everywhere. So there is no bias at this at this level. And there is neither no bias reported for the diffusion, okay? And finally, the insertion of the outer membrane is also achieved thanks to a protein complex, which is called BAM for beta barrel assembly machinery, okay? And again, there is no, so BAM is homogeneously distributed on the outer membrane. And if there is a bias, people have reported that it is more active at the cell center. So meaning that we should not expect at all a polar localization of those proteins. Okay, so and that's why we thought it was it was interesting because when something departs from the null hypothesis, it means that energy should be consumed in order not to observe this null hypothesis. Okay. So, so as I told you, we focused on we focused on one of the adhesion protein. This adhesion protein is called AG43. AG43 is the bacteria adhesion is responsible for bacteria adhesion in E. coli. And so it means that if you have a wild type bacteria, it will express this AG43. It will sedimentate because the bacteria they aggregate together, and on the contrary, in the absence of AG43, there is no sedimentation. Okay. And more generally, Ag43 belongs to the family of autotransporters. And those autotransporters, they are often associated with disease like the dysentery, diarrheas, or urinary tract infection. Uh, and um, and so what we showed using immunofluorescence, so with fixed cells, is that the Ag43 is indeed located at the pole. Okay. So so in order to observe these proteins, it is observing outer membrane protein is not something which is totally trivial. Only a few groups are able to do that, and the reason why this is because we cannot use standard GFP fusion protein to look at these because GFP, if it is folded and fluorescent, it can not go through the sec. So they have changed GFP in order to have a super folded GFP that can cross sec, but then it cannot be taken in charge by the BAM system in order to be inserted. So we design another strategy with colleagues who are chemists and biologists, and this strategy relies on the FAST system. So FAST is a peptide that is twice less the size of GFP that doesn't necessitate a folding. And so this this peptidic tag is not fluorescent by itself, but it becomes fluorescent upon binding with a fluorogen that we put in the environment. Okay, and the good thing with this system is that we can play with the chemical nature of the fluorogen. And, and so by playing with the chemistry of the fluorogen, we can have either non-permeant fluorogen, so meaning that the fluorogen remains on outside the cell, it cannot cross the membranes, or we can use a permeant fluorogen that will cross everything. So and so we tested this, and you see, so these are the three constructions. So here we have the fast floating in the cytoplasm, or fast that is attached to the inner to the cytoplasmic domain of a protein, or the periplasmic domain of another protein. So this is this line. Or the fast is attached to the external, the extracellular domain of truncated antimine. Okay, so this is this line. And you see that the non-permeant fluorogen only reveals this construct which expose fast on the outside of the cell. Okay, so so we use that system in order to monitor Ag43 in E. coli. Surface dynamic, the surface only see the fraction on the outside of the cell, and what you see is that we indeed observe spots that are located at the pole. Okay, and so this is a chimer graph, and you see that during growth, cells the signal gets stronger at the pole. Okay, meaning that adhesion maturates at the pole along the cell cycle. So now, if we move to the periplasmic dynamics, what we see that we have spots, okay, and those spots are moving in the periplasm, okay. So in order to to investigate the dynamics of this spot in more details, what we did is we track those spots. Okay. And so basically, we have a cell mask. We draw the midline of the cell. So we have its length. We normalized all the bacteria so that they have a constant length of 1. Why we do that? Because if we don't do that, we will be dominated by the advection that is due to the insertion of new cell wall material. Okay, so here basically what we question is whether the spots of Ag43 in the periplasm, do they move faster or slower than the growth of cell wall. Okay. And so, and so those who already did single particle tracking know that we quite often end with very short track. This is very difficult to extract diffusion coefficients from that. And so what we did is, so we track the spots, we have a short track, but we circumvent this limitation by studying the bias, the left-right bias at each location of the cell. Okay, so here we have the axis X, which depicts the axial location, and the bias, which I call it delta X. Delta X will be negative if the spot is moving to the left. It will be positive if the spot is moving to the right. Okay, so if it were purely diffusive, we expect a bias along the cell length, which is flat. Okay, and this is not the case. Okay, so here, two things that are important. So first of all, we see spots, but clearly, given so the fast system is pretty good for us, but it comes also with limitation, and one of the limitation is the brightness. So probably the spot that we look at are not a single molecule, but they are aggregates of molecules. Okay. Then, second, the error bar that are depicted here are not the standard deviation. This is because if I plot the standard deviation, they will be big. Okay. And the standard deviation or the variance of this which yield the diffusion coefficient. Okay, and so the system is still dominated by diffusion. So this is the standard error. So how the mean is well defined. And, and so what you see is, so first of all, we have, it seems that there is a bias, okay, because the profile is not flat. The bias finds the center of the cell, okay? And so this, you can see this blue curve as a dynamical system. So here we have fixed point because if a spot is here and the delta X is negative, so it will go to here. And once it has crossed the fixed point, now delta X becomes positive, and so it goes back here, okay? So every time you have a negative slope, the fixed point is stable. And at the stable fixed point, what you see is an accumulation of spots. And on the contrary, we have unstable fixed point. Okay, and for those unstable fixed point, they correspond to a depletion of spot. Okay, so and you see that on the orange curve, which depicts the spot density. Okay. So we were happy with that until we realized that the attraction basin of this central fixed point is larger. So we should have higher concentration of protein at the center rather than on the poles. So we looked at the diffusion coefficient, and we saw that the diffusion coefficient is not homogeneous. Okay, so at the end we have a non-uniform diffusion and a non-uniform advection in the periplasm. Okay, and those two things they give rise to the famous Fokker-Planck equation, for which we can solve the the stationary state and retrieve the effective potential. Okay, and so if we do that, we by injecting the diffusion coefficient and the advection term that we measured, we we have something which is good agreement, meaning that the stationarity hypothesis is is okay. So and so now, sorry, the title is in French. So I saw that Berenice is attending the seminar, so I added those two slides for her. So we then we can question what is the molecular origin of this bias. Okay, and so I skipped details, but we played with membrane potential. We played with cell shape. Okay, and we also played with the mean system. And so the mean system is a system that that exhibits oscillation from pole to pole in the cytoplasm. Okay, and we know that this system is a clue for protein positioning in the cytoplasm. Okay. And so, and so, what we did is to investigate the drift profile in a triple delta mean CDE profile. Okay, and so in this, so I forgot to tell you that the mean CDE system is a diffusion reaction system. Okay, so I can show you after during the discussion exactly what this system is. And so we see that in the triple mutant, there is no more drift, and there is a loss of polarity. Okay. And so, okay, it seems to be okay. We have a positioning system. AG43 responds to this positioning system. But, sorry, this is again in French. So here you see the oscillations. So here, in general, in the literature, the period that is reported for the oscillation is 45 seconds to 1 minute. And this period corresponds to a temperature of 24 degrees and in minimal medium. Okay, so we were able to measure the period of the oscillation in rich medium at 37 degrees, and we found that it is 15 seconds. So it is much faster than the typical division time on the order of 30 minutes. Okay, and so and so the question is how to to relate the density profile of Ag43 in the periplasm to those oscillations. So because there is a clear time scale, a clear separation of time scale between division and the period of the oscillation, we actually consider the mean the mean temporal average profile. Okay, and so this is this is what we get on average for many cells, and you see that there is a good correlation between the two profiles. So everything is fine except that those two systems, okay, they are separated by the inner membrane. Okay, and now what we want to investigate in the future is how the signal is transmitted from one side of the inner membrane to the other side of the inner membrane. And so this is, I come to the conclusion. Sorry, I was long. And so basically, there is, as I hope I convince you, there is a multiscale organization, and so the polarity at single cells organize the cells in the microcolonies. And those organization that favor this organization that favors cell-cell contact then will impact all the population dynamics. And with that, I thank you for your attention. And are the people who contributed to this work.
Thank you, Nicolas, for a very nice talk. So we are still on time, and now we'll stop